Okay, thank you very much for inviting the plant group community to present what we have done. So quickly to introduce the problematic for a plant biologist is to cope with phenomics. In other words, with the combination of environment and genetics that end up with a behavior of the plants. The objective, of course, is to cross data from those three domains, phenomics, genetics, genomics, and environment, both biotic, abiotic, and anthropic. And by anthropic, you've got cultural or agricultural practices. The objective, of course, is to integrate all of those data in a fair manner to be able to do plant breeding, so the observation and the study of genetic variations by traits, or to do climate change studies to study genotype by environment impact. To do this, we gathered in this Plant A Plus demonstrator, and we begin with the obvious constatation that model genomes are available, they are continuously improved, they are shared, we are so far good, and we've got a plethora of phenotypic experiments that produce huge amount of phenotypic data. But we need to be able to bridge those two type of data. And we want to do this within ISBE, ELIXIR, and Asis. And for this, we need, of course, data harmonization. Thankfully, those three infrastructures have been working together for years, also in strong collaboration with the CGIRs, to develop the necessary data standards, and especially MyAPI, the minimum information about a plant phenotyping experiment, and the BRAPI, the breeding API, which is a web service implementation of MyAPI. So what we wanted to do, especially in this Plant A Plus demonstrator, was first to improve the capture of fair metadata. For this, we had two leads. First, working on FAIRDOM-seq to bring plant standards, plant data harmonization means for being able to handle both plant omics, phenomics in this FAIRDOM-seq tool. The other was to work on PISA-3, where the objective was broadly speaking the same. Then we wanted to be able to bring together phenotypic to underlying genomic data using the breeding API. Finally, we wanted to use and to develop tools for the analysis and visualization of data, and especially by using sample Solana assay on maize datasets. So to begin with the first one, the FAIRDOM-seq adaptation to the plant needs. So it's based on Seq for Science. This is a software that has been developed for almost 10 years, I think now. Initially, it was for system biology, but it has been extended to other domain. It's one software with many instances. You've got FAIRDOM Hub, FAIRDOMHub.be, which is still in beta. We've got one at INRA, which is also still in beta, but closing very fast toward production. There is Workflow Hub and so on. The important thing is that FAIRDOM-seq is ISA-based, ISA being for investigation study assay. For us, in the Plant A+, we wanted first to bring MyAPI in FAIRDOM-seq by adding biological material description, by adding phenotyping dataset on the experiments, location, environment metadata, and also by bringing phenotyping ontologies and method, especially observed variables ontologies. We wanted also to enable breeding API in FAIRDOM-seq, since BRAPI is a MyAPI web service, but we decided to delay this implementation because we really wanted to focus on bringing the full MyAPI minimum metadata sets inside FAIRDOM first. So the first thing was about the biological material. It's about the identification of plant genetic resources. And there is a whole section in MyAPI for describing this biological material. It's well it has established. It's an extension of the multi-crop passport descriptor standards that has been used for more than 20 years in plant gene banks. And the good thing is that SIC was in fact already able to cope with this biological material by using the sample type templating system. The only thing that we needed is to improve the documentation, and it's already available, as you can see it, by being able to create new batch submission and new sample batch upload template systems. The second thing was to be able to submit in FAIRDOMSIC batch studies. So many study in with one single series of files. Indeed, whole phenotyping networks, like the one we are handling either within ELIXIR or on Asis, can be several hundreds of studies or even thousands of studies. So of course, it's absolutely impossible to fill the metadata of each of those studies individually. So we've got a close collaboration between this project and the FAIRDOMSIC development team, and to be able to add this new study batch upload button here, which is now, for now, directly very precisely devised for being able to upload MyAPI studies. As you can see, a little bit too fast. As you can see, those studies batch upload allow you with this simple example with only 2 studies to review all of your metadata like your title, description, start and end date, geographical location, and you've got a lot more on the right before finally accepting them. As you can see, with a single file, a single archive, we have the possibility to upload a whole phenotyping investigation in FAIRDOMSIC. That's already a plus because this allows us to connect FAIRDOMSIC with the experimental databases that are in charge of dealing with day-by-day experimentation and therefore enabling later integration with genotyping data. The other thing that we needed to do is to be able to cope with custom metadata. Indeed, MyAPI is a minimum information about plant phenotyping experiments, but nonetheless, we introduced between 50 and 80 new fields. If the FAIRDOMSIC team was to add all the fields requested by all communities, they would very quickly end up with a lot of trouble. Therefore, they developed, with our contribution, a custom metadata system that has the possibility to add any metadata scheme. Of course, the default configuration is for MyAPI investigation and study. And as you can see, we have the licensed MyAPI versions that are added to investigation, the study start date, end date, latitude, longitude, etc., added to study, etc. And it is fully configurable with other scheme in the future. And now we will go on with PISA. So Kristina, if you want to talk on, just let me know when I change slides.
Thank you, Cyril. Yes, I will call you when the slides needs to be moved. So can you hear me well? Is it okay?
Yes.
Okay. The next tool that we were developing is PISA-3 tool, and it's kind of complementary to the MyAPI tool that was just described by Cyril, and it's meant mainly for smaller labs that maybe do not have the capacity to build up the whole SIG system at their own institute. So it's It's very easy, but it allows communication with all SIG platforms, so it's totally complementary. And additionally, we also developed some metadata formats that were still missing from SIG formats. So Cyril, can you please move? As I said, it's very simple. It's a system of batch files that creates standard directory tree, which is aligned with ISATEP format. So we have the project level, investigation level, study level, and the assay level. And each level is then prescribed with certain metadata requirements that are in line with everything that is in SIG platform, except for the specific ones at the assay level that describe certain experiments. As I said, we developed, introduced all the metadata on the assay level based on the SIGGERM templates, but for some they were missing. And for example, for RNA-seq mapping and for statistics analysis, we developed new templates for metadata, similarly for promoter activation assay, qPCR quantification. And as Cyril described, we also worked quite a lot on MyAPI sample descriptions, which are then also, in our case, aligned with Cyril's approach, described on the study level, and then you can make different sub-analysis on the assay level of these samples. So if you can go to the next slide, please. So this is just a snapshot from our PISA project system on server. So you can see that we are really running it on many projects now, and also it is already implemented in at least 5 groups across Europe. This is the insight into one of the projects at the investigation level, and you see that you have another batch file to make another investigation, and certain folder structure then appears with certain metadata requirements. And similarly, then if you go into the investigation, you can create new studies and so on and so forth, also for the assay level. Okay, for the next slide, I think that now it's back. Okay, very important thing. that the part of the being fair is also that the data are findable and available. So we, in collaboration with the Fairdom Hub SIG team, we designed a system for automatic upload of everything that we have in PISA tree tool to Fairdom Hub. So they developed the API, and we developed an R script named SIG-R, and it's really working very nicely. There were some problems with synchronization because we allowed more metadata files on different levels. This was still not allowed in SIG, but yeah, we are working on that still. Okay, I think that is all, and then it's back to you, Cyril.
So to answer the questions, the future work, sustainability on project, on what are we going to do now the project is done. So the first thing I'd like to underline on the Fairdom SIG part is that the custom metadata on most of the work that we have done in collaboration is now merged in the main version of Fairdom SIG. Therefore, it will be included in the next official release of Fairdom SIG. We are actively collaborating right now on adding more MyAPI metadata, mainly the observed variables that follow the crop ontology triplet model. Indeed, for describing the measurement procedure with plant phenotyping, we needed a slightly more complicated ontology model than usual, with each phenotyping variable being a combination of one trait plus one method plus one scale or unit. So this is active work in progress. We are also using Fairdom SIG as a prototype service on ELIXIR nodes on H2020 project. So Belgium is using it a lot. French side, we are also using it very actively. And I'm pretty sure there are other groups interested. And finally, we will need a high level of trainings, webinars, datathons that are currently planned within the ELIXIR nodes and the H2020 project for tools and good practice adoption and improvement. Considering the impact, we've got efficient solutions for solving the first mile of bringing data toward general availability. We've got broker formatter candidate solution for preparing data submission for phenotyping repositories, whether it is Dataverse-based solutions such as datainray.fr, GNPIS, or EDAR. I'd like to thanks very much and to point the complementarity and the very good collaboration with the FAIRDOM-SIG teams. There are interesting perspectives with emphasis databases. And globally, for me, it's really a success. And we have made also extensive use of the biohackathon for the past 2 years. And we now go to the last deliverable of the Plant A+ project with the gene expression plotter. And Asis,
Okay, thanks.
for you now.
I will, due to the advanced time, I will skip the slides. Basically, we developed a tool to visualize gene expression data, which is one form of phenotypic data, as it is the genetic response of a plant to certain experimental conditions. With I want to share my screen. Oh, I cannot share it. I wanted to do a live demonstration.
I'm stopping the screen sharing so you can share yours.
Okay. Okay. Can you see my screen?
Yes.
Okay. So this is the tool. It is online. We use GitHub IO pages to host it. You see there's a fair amount of different methods to import your data. You can use the tables as they are provided by the standard RNA-seq analysis tools like Callisto, Salmon, HiSat, and so on. There is an import for Excel tables that you can provide yourself. They are fairly simple. And we also introduced a link directly to the FAIRDOM Hub data. We actually used potato virus Y infection experiment from Kristina's group. And upon clicking that link, data will be downloaded from here, namely the metadata and the normalized counts. The data will be parsed, and then you have the data right in your browser and can start plotting away. For example, this is a legend that I prepared for it. So we abbreviated the different groups that refer to a certain combination of genotype, treatment, leaf number, and leaf tissue. So then you can add a new plot and, for example, add this array locus, select where you want it in curves or bars, and then you can see the expression of the gene here with a legend, and compare the expression of the gene within different groups. You can also use several genes in one plot and then compare the expression of the genes in the respective groups, like so. And there is a third version where you can use curves with that. You can just start typing your gene name. Oh, this one is not in there. That's actually that one. And here you can see two different curves for the two different microarray locations, comparing them into different groups. You can see the green one is in most cases more highly expressed than the red one. And because this tool is generic, you can integrate data from any source. And we have a standardized upload format, which is this table. You can just use it to visualize your gene expression data. And there is a complete guide and an API. You can even use this within your own laboratory to pre-deploy it with your custom data and switch off all this data loading and share it in your group to visualize your gene expression project. And I think that's basically it.